In the last decade, the ability to have genetic targeting of cell types in mice and to be able to manipulate specific cell types, work out their connectivity, has really provided huge insights into the way the brain works. And there's going to come a day, it might be 30 years or 50 years from now, where there will be gene therapy in the human brain targeting cell types and manipulating circuits that are defective in brain disorders.
So Ed Callaway, it's we never met in person before this, but it's really, I mean, having read your papers over the years, it's really great to have you here in Rochester.
Yeah.
And wanted to introduce you to our community and ask you a few questions about your science and how you ended up where you're at in the world. So you're at the Salk Institute, and have you spent your entire career out on the West Coast?
Well, my first faculty position for 3 years, I was in Colorado at the Health Sciences Center in Denver, and then moved in 1995 to Salk Institute. So it's been 24 years there now.
Excellent. Yeah. Well, let's dive into your science. I of course, I was reading beforehand, trying to get ready to meet with you, and I got very struck by something that was on one of the sets of materials, and it was describing the brain as a veritable bowl of spaghetti, and so a lot of your work has been about untangling the mysteries of the circuitry of the brain, and would you like to tell us a little bit about how you think about brain circuitry and this mass of neurons and interconnectivity?
Well, there are two levels of different ways you can entangle that. And the one that I think is the most tractable right now and where the field is really moving is cell types, right? Because even though it's a big tangle, if we can, label one of those pieces of spaghetti and follow it, or it's maybe not just a big pile of spaghetti, it's got some penne pasta and rigatoni and different kind of things in it. But without having the ability to target and see that that's all in there and each of those is different, it's pretty clear to us now that cell types matter. Each cell type is connected in a different way, mediates brain function in different ways. And in the last decade, the ability to have genetic targeting of cell types in mice and to be able to manipulate specific cell types, work out their connectivity, has really provided huge insights into the way the brain works. And there's going to come a day, it might be 30 years or 50 years from now, where there will be gene therapy in the human brain targeting cell types and manipulating circuits that are defective in brain disorders.
Is it the idea then that there may be disorders that are allied to specific deficits in specific cell types? That's why we need to know.
Yeah, and there's evidence for that. So, for example, postmortem tissue from schizophrenic patients, there's a particular cell type called chandelier cells. They make a particular kind of synapse on pyramidal cell axon terminals, beautiful chandelier-like axons. That's how they got their name from the old days when you've Golgi labeling of neurons. It turns out in schizophrenic patients, they have fewer than normal number of these axon terminals on the pyramidal neurons. So that's just a clue that there might be something wrong there, but there now exist mouse lines that you can target gene expression to just the chandelier cells. And you can inactivate and activate them and see what their role is in the function of a mouse brain. And within a couple of years, there will be ways to target chandelier cells in a monkey brain, which is much closer to a human brain in its function, and where you can study higher level cognitive functions and manipulate them and look at them in that context of something. And
so now you're talking about looking at these cells, and of course people.
I'm sort of looking in a figurative sense.
Yeah, I know, right?
So we do look at them.
Exactly, and that's in some ways that's where your lab has really become famous around that. So there are ways to stain these neurons, but you brought new technologies using a virus, right? A very famous virus to bear in terms of our ability to really paint these pictures. And would you want to talk about that?
Yeah, sure. Well, for many years we wanted to have a way of, if all these cell types are mixed together and you want to untangle the connectivity, you'd like to know where are all the cells in the brain that connect to a particular cell type in a particular place, that starts in a particular place in the brain. And back, I guess it's been about 2007 now, about 12 years ago, we first developed this method, working very closely with Ian Wickersham, who was a graduate student in the lab. We modified rabies virus, which has natural abilities to spread very selectively only across connected neurons. but it doesn't have a property where you'd inject it in the brain and it would only infect a cell type. And it doesn't have the property where it would spread one step and stop, so you could unambiguously say, those are the cells connected. It would just keep spreading. And in fact, in animals that are infected, like bats, they'll eventually change their behavior and they'll spread to other bats and they die. But we modified the virus in ways that allowed us to target its infection to specific cell types, and so that it would only spread one synaptic step and label the direct inputs to those cells. Then you can look across the entire brain and look at those. And you can even do functional studies. You have people here in Rochester that are taking advantage of this virus to express optogenetic constructs which allow you to turn those cells that are connected in specific ways on and off to look at their roles in function in the intact brain. So,
and
yeah.
15 years ago, if somebody had said to you it would be possible to switch on and off cells with light that are transfected with the rabies virus, would you have laughed at them, or did you see it coming?
I mean, optogenetics has been around, probably, is it getting close to 15 years? I'm not sure. Yeah. And we worked on other methods to inactivate that were chemical, because we did want to be able to target cell types in brains of animals like monkeys. We're only just now getting the ability to target cell types in monkeys very selectively. It's something we imagined 15 years ago. I think the first experiments that got us thinking about tracing connections in cell type specific ways were ones that used a different virus called PRV. It's in the herpes family of viruses, like chickenpox viruses and things like that. Jeff Friedman and Lynn Enquist made a version of that could label the inputs to a specific cell type, but it kept spreading multiple synaptic contacts. And we actually worked with that for a couple years trying to get a way to make it stop. But all those things didn't work, and it was those failures that led us to the properties of rabies virus as one that we would be able to engineer in a way that would work. That was more than 15 years ago. They published that paper in 2001 showing that tracing.
So all this work, it led to quite an honor for you this past year, right? Where I believe you were inducted into the National Academy of Sciences, which is our most prestigious scientific society. How did you find out? Did you get a phone call? Did they send you a letter?
That's how it usually works, apparently, is they have this thing of, especially if you're on the West Coast, and it's early in the morning. It must have, well, to me early, I would sleep late, 8 in the morning. But I actually found out from my daughter, strangely, because I always silence my cell phone when I go to bed, and I'd gotten up and was in the shower, and I didn't notice that people had called and left messages. And then I opened my computer to check traffic in the morning. I happened to look at my emails and see all these sort of congratulations messages. And I was like, oh, what's that for, I wonder? Right. And then next thing I know, my phone's ringing and it's my daughter telling me. So
fantastic.
Yeah. So, and then of course I called and talked to the
and tell us, how did it feel? Were you surprised?
Well,
or was
because I mean, it's not something I was really on my radar thinking about, and they do have a meeting every year at a certain time. I didn't know this was the time when the meeting was happening and they would be doing this voting. Of course, you have a sense that you're being nominated because the people nominating you have to ask for materials. They can't tell you they're nominating you, but you can tell from the format of it. Right. It's a nice honor, and really mostly a reflection of all the stuff that people in my lab over the years have done.
Here you are, a National Academy member, but let's go back in time and track. How do you end up as a National Academy member? Tell us about the academic trajectory, and when did you figure out you wanted to be a neuroscientist? Was this something when you were 10 years of age, or was it a later?
Well, I mean, I've always, even since when I was a kid, fascinated with how things work, and sort of, would take apart mechanical things, like to work on bikes and cars and things like that, and like to build things, which helps a lot with neuroscience because you have to, especially longer ago, build all of your own things, but just really a fascination with how things work. But it wasn't until I was in college. I knew I was interested in biology and was dabbling with the idea of medical school. But during my sophomore year in college is when I first started the first biology lectures. First years you take chemistry, physics, math, and learned about neurons and that they have this electrical activity and action potentials. These beautiful lectures that Corey Goodman gave. He was a assistant professor at Stanford at the time. That's like 30 years ago. And that was when it just hit me. This is what makes me who I am. Everything about me and how I work and think and feel is because of these neurons in the brain and their activity together. How does that work?
Right.
And what could be more fascinating? So I just started at that time signing up for every neuroscience class I could find, and found a lab to work in with Jack McMahon, and studied neuromuscular development and got interested in nervous system development. Yeah.
Now, I happen to know you and I have something in common. I don't think you know this because I think we both started out life as dumb jocks. Did you end up at university?
I hope I wasn't dumb. Yeah. I mean, Stanford has some academic standards, but I do think they are a little relaxed if you're a fast runner, because I might not have gotten in if I wasn't a fast runner, but that led to all kinds of opportunities. Yeah. So it kind of goes with being a scientist. I mean, distance runners are a bit obsessive, and you have to just run a long time and for what might be a reward that may or may not happen later to win. But I was very competitive, and so it's a sport where hard work pays off, and I think that's true of science too. And I think I learned a lot of lessons through that, and also being an athlete in college, you have to be very efficient with how you use your time, because I mean, you're taking a full course load, and you've got to work out every day, and all your friends are out, laying on the lawn in the sun or playing volleyball or something for fun, and you're out working hard, running, and come back tired and still have to go to the library and study. So I think it taught a lot of self-discipline, and I love just the camaraderie of that.
Right.
And that's something that the lab also does for you, right? That you have this group of people that you work with every day.
That are almost family because of the closeness and the intensity of it, the engagement.
And the graduate students and postdocs, they're putting their career in your hands, and you got to, look out for them. And
right. So I didn't mention that before, but I came to the United States on a track and field scholarship.
Oh, really?
Brought the same kind of thing. And I also believe that there's this sort of obsessional aspect to folks in the sciences, and you find out when you dig a little deeply that they've run track and field at a high level, or they've played a musical instrument at a high level, or they're a very fine artist as well. And there's a little bit of the manic. Musicians
are great to work in the lab because they do the same thing over and over and practice.
Exactly, and that's and the delayed gratification, the part where you need to stay applied to something for hours and hours and hours with very little return, but the long-term goal. Very good. You have 3 children, and I was fascinated by this some more from a human interest perspective, because they've grown up in a household where their dad is a very, very prominent scientist. Are any of them following in your footsteps, or are they steering clear of it?
One of them is. I mean, mostly they have remained. It's good to be kind of oblivious to exactly everything we're doing. And I mean, you're just their dad. What's the difference as scientists? So I have, with my 3 children, the oldest is a computer science engineering guy, and of course works in the Bay Area and does very well in that. My older daughter is in grad school at Berkeley in immunology, virology, and she was the one who paid the most attention, especially during the early rabies days, and I gave talks at her school about rabies virus stuff, and maybe that influenced her interest in virology and diseases. So she studies childhood malaria in a lab at UC San Francisco and through a graduate program at Berkeley. And then my youngest daughter is applying to law school this year and working in the public defender's office in Oakland. So they're all doing great things, very proud of all of them.
Yeah. So what about work-life balance? And all through the years when you're raising children, I think people are often think about scientists and the quantity of hours that have to be put in the lab, and I think it's a reality of the world we live in.
And especially in the earlier years, I spent a lot more time in the lab, and I'm very fortunate that my wife was very supportive of that. And I also traveled more than I do now at times, and that's really helpful to have a partner who helps you to manage all that and tolerates it to a certain degree too, because there were times when we did all-night experiments and things. I do think you have to be able to turn it off at times too, and when you're done at the end of the day, to be able to come home. And that was when the kids were younger. One of my favorite things was it was time to go home, give everybody a bath, and read.
Yeah.
and I would read them books.
And what about some advice? So, a young graduate student thinking about life as a scientist today, 2019, going forward, the challenges of it. The young graduate student thinking about having a family in this field and in this business. Do you have any pearls of wisdom?
Yeah, I mean, it's nothing that's not common sense. You have to make the choice that works for you. I mean, when I was in grad school, I was very focused. I probably was in the lab 12 to 16 hours a day and didn't really have a lot of balance, but didn't have any kids yet either. And so I consciously, my wife and I discussed that we should wait till I'm a postdoc to have our first kid. But then our first kid, our son, was born while I was a postdoc, and that worked out just fine. Everything's hard. No matter where you do it, there's no easy time. And but if you believe in what you're doing and you like it, it's difficult, but it's not hard in the sense that you're enjoying what you're doing.
Right. Is there a difference for youngsters these days? When maybe you and I were coming up through the system, you became an expert in a very specific domain, and today you really have to start to work across disciplines.
It's harder for sure, and in neuroscience now in particular, because what you could possibly do with the range of tools that has emerged, the expectation for a high-profile paper is that it's going to have a little bit of everything. And how could you do all that? So I think it becomes more important to work as a team and to have different people in the group that, you see more co-first author or even three all gave the same amount of effort to the paper going on. But it's also good to learn from the other people that you're working with, because eventually you do need to go start your own lab, and it's good to have learned multiple skills and bring that and to be able to train the people that are coming in.
Well, can I lean on that? Eventually you have to go start your own lab. Are our models correct
you get
today?
To. You don't have to.
Right.
So you get the opportunity.
Yeah. But are we doing? So would it be fair? I mean, you can push back on this. Would it be fair to say that the way we do business today, with a new investigator gets a job in a lab at a university, opens up their lab and starts, it hasn't changed much over the last 40 or 50 years. We still operate under the same basic model. Is that the right way to do science these days? If you were shaking it up, would you approach it in a different way?
Perhaps. I think it probably depends on the kinds of institution and a lot of institutions have a really important emphasis on teaching as well, and other things that need to be done. A really good model of doing it a different way is the Allen Brain Institute, right? And they have a very different structure where they bring in people. They actually pay people at postdoc level a lot more than our postdocs get paid. We should pay our postdocs a lot more. But that's dictated largely by government levels that are set. So,
so what's the model there for our audience?
Much more of team kind of science, and so they do have people who are more specialized and have careers that are doing science, and some of those people move through the ranks and become the people who are more interested in asking big questions and directing programs. Other people would rather stay in what they're doing, but they can get paid a good wage, often better than what some starting assistant professors do, and have a career in that.
As a career scientist, as part of
working as somebody who does bioinformatics, or somebody who does slice physiology, or does calcium imaging in vivo, or does electron microscopy, and all these different things. Because that's one of the real inefficiencies in the way the things work now, is that I have somebody who comes as a postdoc, and just as they get really good at what they're doing, they leave, and I have to replace them with somebody else that's going to come in and start over.
All that training.
And we do often have sort of career research assistants who stay in the lab, but the system is much more designed to have funding for postdocs and grad students through fellowships and different kind of things. So we rely on that labor, I think, more than we should.
Right.
There are other models in other countries where they give you a budget and say, hey, this is for technicians, and then here's the budget for these people. But here it works differently. So I think that, it's kind of like how things are in the U.S. It's not the perfect system, but it works well, and we do well with it. You could design a better system, but then if you try to impose things, then you have unintended consequences at times too.
So looking to the future now, you mentioned, specific cell type targeting and genetic manipulation of specific cell types as ways to get after disease. But looking out 10 years, 15 years from now, what's on the horizon? What do you see as the really exciting areas that we should be pursuing?
Well, in terms of targeting cell types, I think that the field is just now it's probably been in the last year, but what I'm seeing in publications, and there's more and more of a trend to people to publish preprints on bioRxiv. And just in the last 6 months, there have been what people call enhancers, cell type specific regulatory elements in the genome that are responsible for the normal mechanisms that give rise to the differences between cell types. Cell types are different, not because they have different genes in them. They all have the same genetic material, but it's which genes get turned on and which genes turned off during development and the specification of a cell to a type. And there are marks of that in the genome, and there are analyses that can be done called epigenetic analysis that can look at those marks in the genome and find out where are these elements that might be enhancer elements in the genome that might be responsible for the difference between this cell and this cell and this cell. And there are some of these publications now that are coming out showing that you can use that analysis to predict enhancers. You can put them in a viral vector, the same viruses that are used now for gene therapy in humans. And you can then, in fact, even human tissue, in one case from the Allen Institute, where they have tissue from people that are having tumor surgery or epilepsy surgeries, and those patients donate some of their brain that can then be cultured, and you can test those viruses, and they've shown that you can get the targeting to the cell type that was intended based on their epigenetic analysis of these marks. And I think that in the next just couple years, there's going to be a huge explosion in our ability to target gene expression to cell types, not just in mice where we have to genetically engineer the genome of the mouse, but where we can go into any species, including humans, and target a cell type for therapeutic purposes.
A great future, and it's on the horizon here. It's not a million miles away at all.
Yeah, and I don't think it's overhyped at all. Yeah. Now, how long is it going to be till you actually do gene therapy in a human? You're going to have that would require cell type targeting. There will probably be types of diseases that are more tractable, and the cost-benefit kind of equations where, if maybe somebody who has epilepsy, the treatment now is to remove that part of the brain. So there's not a huge cost to saying, well, we have a potential gene therapy.
First before we can take it out.
Yeah, we can do something where we would express a transgene, only in a cell type that would then, if you activated that cell type, would suppress the activity. And then if you detected a seizure coming, you could maybe do that. Or if you had a drug, the transgene is affected by a drug that the person takes that's targeted only to those cells rather than the drug that would have all kinds of side effects for the epilepsy drug somebody might be taking now. You've now targeted it to just that part of the brain where they have the epileptic focus, and you can just regulate with the dose of this drug, which doesn't have any side effects because there are no receptors for it in the brain. So that's something that you could imagine coming along. And you start small with something that's cost benefit is big, because if it doesn't work or something goes wrong, you're just going to cut that tissue out anyway. Right. But then building to things that are more sophisticated over the years. I think the basic research is necessary to identify how the normal circuit mechanisms work, and then to be able to say there is a target, there's a treatment that you might try. And there's going to have to be translation across species. You're not going to go straight from mice to humans. Right. It's very clear now that you can find homologous cell types, 100 cell types in the cerebral cortex of people and mice, and you can find nearly all of them are homologous. But the genes that are expressed in the primate cell types, in monkeys and chimps and humans, that's highly conserved, but it's different in mice. Right. So the way we're going to target those cell types and the genes and how they relate to genetic disorders
Right.
and genetic prevalence for disorders is going to be different between mice and monkeys. You need a transitional species to work with to do that.
So we're getting close on our time. I wanted to ask you, when at the end of the day, I assume these days you're not running cross country to wind down. How do you switch off? What's your thing?
Well, the last few months haven't been as diligent, but I do still try to run a few days a week. Go for a 5 or 6 mile run. I like going hiking. I like going fishing.
Yeah. Do you find, like I do, when you're running, that there's a clarity? It's a really good time to think about science, actually, in a
usually when you're not thinking about it. Yeah.
Yeah.
That happens sometimes. You just have a chance to go.
I often wonder about the
head.
neurotransmitter milieu that allows for that kind of clarity when you're on a nice. No, they're not all nice. Sometimes it's just a slog, but when the mind clears on a run and all the rest of the world falls away, sometimes I have some of my best thoughts about experiments. And it's such a pleasure to have you here in Rochester.
Thank you.
I really
I've
appreciate you
enjoyed it.
coming in. Excellent. Thank you.